DNA polymerases. There are different types of DNA polymerases which are used during the PCR reaction. The most common DNA polymerases, they are tag polymerase. Tag polymerase extension rate is 75 nucleotides per second, and this polymerase was purified from Thermus aquaticus. The second type of the DNA polymerase is called as ampli-tag, which is also called as Stoffel fragment. The extension rate of this DNA polymerase is approximately 50 nucleotides per second, and this polymerase was also purified from Thermus aquaticus and later on commercially produced. The third type of the DNA polymerase is called as Vent, and its extension rate is approximately 80 nucleotides per second, and this DNA polymerase was purified from Thermococcus litoralis. The fourth type of the polymerase is called as PFU, and its extension rate is 60 nucleotides per second, and this polymerase was purified from Pyrococcus furiosus. There is another type of the DNA polymerase which is called as TTH, and its extension rate is approximately 33 nucleotides per second, and this DNA polymerase was purified from Thermus thermophilus. So these are different types of the DNA polymerases which are used for the PCR reaction. Out of all these polymerases, the most common DNA polymerase is called tag polymerase. So this is one of the most commonly used tag polymerase. And in some PCR reactions, like if we say in case of long PCR, there can be mix of different DNA polymerases. So different DNA polymerases, they are combinedly used in case of long PCR. And in case of reverse transcriptase, mostly TTH, that is a polymerase purified from Thermus thermophilus, is used for the reverse transcriptase activity. Here we can see that how DNA polymerase extend the newly synthesized strand. DNA is double stranded. Here we can see that DNA is double stranded. This is strand number 1. Let's say this is strand number 2. Here DNA has been denatured, and both strands, they are separate from each other. Here we can see activity of the DNA polymerase on this strand, which is the leading strand, and here we can see the activity of the DNA polymerase on the lagging strand. So here we can see that DNA polymerase is extending the newly synthesized strand. So DNA polymerase is actually the main component in case of PCR reaction, because it extends or it forms new strands by incorporating the dNTPs. Common polymerases and their origin, which is also called as tag polymerase, was purified from first time from Thermus aquaticus, then TFL, that is Thermus flavus, then TTH, Thermus thermophilus, TLI, Thermococcus litoralis, and PFU, that is from Pyrococcus furiosus. So these are common polymerases and from the source where they were purified. So we can say that most common types of the DNA polymerase, they are tag polymerase, PFU polymerase, and Vent polymerase. These are some of the most commonly used DNA polymerases in PCR reactions.